Okay, we are now going to talk about something called weak terminators. And they're a lot like regular terminators. We learned the terminator structure, and I drew it kind of like this. All right, so these are going to stick to these, and you're going to produce this structure. Now, generally, a strong one, when this is, complementary to this, and it's perfect, it's a perfect complement, we're going to get what we call a strong terminator. All right, because all of these will stick to all of these, and none of these stick to any of these. So that's a strong terminator. But what if we were to replace a few of these? Maybe we turn that into a C, and that one into an A, and that one, but we could turn that into a T. That would still be complement. But let's see, we'll turn that one into a T. So now we would get A, T, G, C, A, T, T, C. That actually would still stick pretty well. It's really just this one and this one. I guess the end one doesn't stick. So we've reduced the amount of stickability. We've reduced the amount of stability is the word that we want to use. So we would call this a weak terminator. All right. And this has implications for what we're going to talk about. And these weak terminators, they kind of go between here and here, back and forth. The strong ones, once they form, they pretty much stay there. These, they kind of transition back and forth. They'll pop up into this form, and then spontaneously, every now and then, they'll go back here. They'll stay there a while, and then go back to this one, and back and forth, back and forth. And so the weak terminators kind of are there sometimes, and sometimes they're not. And the cell utilizes these for a very specific function, and that's to control ratios of the proteins. So ratios are what we're talking about now. So let me go ahead and start a new picture here. Let me get up here. And let's build our, at least part of our operon again. And over here, let's build our strong terminator at the end that we know about. We'll put our data box here. These are the only parts we're worried about. We're going to put in Z, we'll put in Y, and we'll put in A. And then we're going to draw these with dashed lines. Like that, to mean they're weak terminators. So here are our two weak terminators. It's kind of interesting where you put them. And what this means is that, all right, so the RNA polymerase sticks and it starts moving across here. Well, every now and then, it's going to run into one of these. So let's say that this is here. Well, let's make it a little stronger. Let's make it say that it's here 9 out of 10 times. And that this one is here 9 out of 10 times. Well, what that means, if it's that strong, is that this will get here all the time, all right? 10 times. It's going to go through here 10 times. And 10 of those times, this thing is going to be there, all right? So for 9 of those times, it's going to be strong, be up. And only 1 is it going to be flat and get through. Well, that means we come down here and it gets through here 9 times, it's going to keep going. It's going to get to here. All right. I'm sorry. I should have put that down here. It's flat down here. So this one, let me undo. All right. There we go. So remember, it always makes it to here. But one time it's going to make it through, 9 times it's going to stop right there because it was flat. All right, so that means it's going to keep going. All right, so here it has switched. Well, same over here. One, it doesn't get to here, but every now and then it will get to here and it will just stop. And all you're going to make, so here 9 times we made this, and then 1 time out of that 9 we made this. Well, if we increase this and do it 100, well, every now and then if you did it, it would get through both of them, and both of them would be flat. And if both of them are flat, then you would get to A. All right, and then here's your strong terminator here. Weak and weak. And if you add up the numbers that I put up here with this, it means that if you did this 100 times, well, you always, if you look at what you made on this RNA, you always get Z, and you'll notice Z is always made. So you're going to make, oops, I should have 100. So you're always going to make Z. But only sometimes are you going to get over here to, out of that 100, you're only going to get to this one 1 out of every 10 times. So you're only going to make 10 Y's. Okay? And then out of that, out of these 10, you're only going to make 1 A. You always make it to Z. 1 out of every 10 times you're going to make it through Y. And then only 1 out of 100 are you going to make it through both Y, both of these weak terminators, and get all the way to A. So what this does, or what I'm saying, is that these weak terminators, all right, because they go flat sometimes, they allow the cell to control how many of these are made if the operon is on. Of course, this has to be on so that the polymerase can be through here. And so when it goes through here, it's always going to make Z. And Z, that makes sense, because you need a lot of Z. You've got your cell, and you got all this all this lactose in here. You got to have a lot of the enzyme to break it down. So one enzyme, one of these Z enzymes, the Z protein, that chop up the lactose into glucose and maltose. You need a lot of those because you have a lot of lactose in there. But you don't need many of the poor proteins that let the lactose through, because it's going to let a lot of the lactose in. Just make one, and you can, given time, it would let it all in. So you make a few of them, and it's going to let the lactose in, and you're going to be fine. You certainly don't have to make as many of these as you made of these. And then the A detoxifies things that sneak in. And you don't have to make a lot of those. You just have to make enough so that it can kind of go around and be the security guard. And if there's something in there that shouldn't be in there, it can detoxify it. So you don't have to make many of these. A is especially expensive. So you don't want to make many of these unless you have to. And so this actually fits in perfect with the idea of the cell conserving its energy. Not only is it only turning on ZYA when lactose is present, it also only makes the amount of Z, Y, and A that are needed for their function, so that you don't have what? You don't have too much Z, all right, or Y, or A, given the function that it has. You only need a few of these, you only need a few more of these, and you need a lot of this. So the order here is telling you which one you need to make more of. You need to make a lot of this. This is important for doing the digestion. All right, this is for letting it in. You don't need as many of these because you only need so many doors or openings to let many lactoses in. And then you only need a few of these just to make sure if something sneaks in that you have a way to deal with it. And this controls the ratio. So the ratio of Z, Y, and A are controlled by these weak terminators. So understand that the ratio is controlled by weak terminators. Another method for the bacteria to conserve energy and resources that might otherwise go towards something else if this didn't work. All right, and we will look for these as well. You can find these just like you look for the O's. There are ways to use dot plot to find these terminators. I actually haven't shown you that yet, but you can find a terminator. Strong ones are easy. The weaker ones, not so much, but they're findable, and we'll learn how to do that. And you'll have to annotate the genome, the lac operon, and we'll find these together. And if they're present, perhaps, in the operon I give you, you'll need to find them. You'll need to at least look for them. All right, that should do it for this video.